Welcome to the March edition of AJP-Heart and Circulatory Physiology video table of contents, or from the editor's desk. I think you'll find the papers this month especially interesting, with something for everyone. We've published one review, 24 original research papers, including one rapid report and one editorial focus piece. Here are some of the highlights. As part of our call for papers on cardiac regeneration and repair, Katz et al. Provide a review summarizing the important role of microRNA as regulators of cardiomyocyte differentiation and proliferation. These authors summarize the relevant processes and recent progress regarding the function of specific microRNAs in cardiac development and regeneration. This is a very timely review that should be of interest to all. Gillis et al. Studied diaphragmatic function in two models of heart failure, the TAC and MI model. 18 weeks after surgery, in vitro diaphragmatic force was preserved. However, calcium-force relationships were significantly altered, including myofilament phosphorylation. There were also shifts in myosin isoforms and the phosphorylation of troponin T, desmin, and myosin light chain. These results indicate that there are multiple changes in diaphragmatic myofilament function specific to the type and stage of heart failure and occur before overt impact of in vitro force production. Astronaut Scott Kelly just returned to Earth after 1 year in space. In a timely study by Usin et al., carotid arterial stiffness was measured in male and female astronauts within 38 hours of return to Earth after spending 6 months in space. Carotid artery distensibility and stiffness indices increased by 17 to 30%, which was greater in females, but pulse wave transit time was greater in men. Blood biomarkers showed an increase in insulin resistance greater in men, but renin and aldosterone greater in women. The authors conclude that altered hydrostatic arterial pressure gradients, as well as changes in insulin resistance, may contribute to changes in arterial properties, and these mechanisms may be gender-specific. Consavallis et al. Studied the impact of a Western diet on biventricular structure and function, as well as endocrine and metabolic changes before overt obesity. Rats were compared after 6 weeks on a normal diet or an increased caloric intake diet. Over this period of time, the Western diet animals showed increased adiposity but had similar body weights. Cardiac abnormalities included biventricular hypertrophy, increased stiffness, and impaired relaxation. Adipose tissue exhibited a pro-inflammatory environment. Evidence is also provided to implicate galactin-3, a pro-fibrotic molecule, as a biomarker before overt obesity is established. Hamilton et al. Tested the effects of combined L-NAME and angiotensin II infusion on cardiac metabolism and gene expression in mice over a 5-week period. Compared to the agents given individually, together evoked the most severe heart failure and increase in oxidative stress. There was profound reduction in mitochondrial function as well as reduced respiratory ratios in subsarcolemmal mitochondria and reduced state 3 levels for complex 1 substrates. Pathway analysis indicated significant changes in metabolic pathways such as oxidative phosphorylation, mitochondrial function, cardiac hypertrophy, and fatty acid metabolism. Finally, Goodwill et al. Examined the hypothesis that Kv7 channels are present in cells of the coronary vasculature and regulate vasodilation in swine. Coronary arteries expressed KCNQ gene and the Kv7.4 protein. Flupirtine, a Kv7 opener, relaxed coronary artery rings, and this was attenuated by linopridine, a Kv7 blocker. Bradykinin-induced relaxation was inhibited by linopridine. This finding was replicated in vivo. These data indicate that Kv7 channels are expressed in some coronary cell types and influence endothelial function. Thank you for watching another edition of AJP Heart and Circulatory Physiology video table of contents. I hope to see you next month, and if you run into me at experimental biology, please say hello. I approve this message because we publish great cardiovascular science.